Good morning. Thank you for coming for presentation by Dr. Christopher Peterson. He is an assistant professor in the Translational Science and Therapeutics Division in Hutchinson Cancer Center, as well as University of Washington from Seattle. He's been developing cell and gene therapy strategies, and he's our collaborator for our NIH-funded U19 project with Dr. Scott Kitchen, Koki Morizono, as well as Angie and Irvin. So he has a tremendous contribution for HIV research and try to cure HIV by modifying hematopoietic stem cells with many different anti-HIV genes. In particular, he's going to talk about his research on modifying stem cells with CAR T cells and how they can improve the system for gene modification and stimulation in vivo. Particularly, his expertise is using non-human primates, which is a very precious model, the closest model for modeling human clinic. So I am very excited to bring him to UCLA. And please welcome Dr. Christopher Peterson. Thank you.
Thank you very much, Dong-Sung. Thank you all for coming. I've had a lot of fun. This is my first time to the UCLA campus. It's beautiful and much better weather than Seattle, where I'm coming from. So I'm going to talk to you today about a few stories, and I've just started my own laboratory at the Fred Hutch in Seattle. So I'm going to talk about some of the stuff that we're just getting going with. So this is where the story begins, and this is the major problem that we're trying to overcome, which is that latently infected cells in people living with HIV essentially persist lifelong. So this is a schematic from a review from a long time ago now, but is still highly relevant, that when a person becomes infected with HIV, there are a number of different cell types that are infected. They're shown here in different colors. And what you can see is that some of these cell types are short-lived, like the yellow and green and gray. And some other cell types that are infected are very long-lived. And there's increasing amounts of data that these cells essentially live for the entire lifetime of the individual. So we have to actively find these cells that carry the HIV provirus and clear them out of the body. And that's not just in blood. I think that's one of the most important things to highlight here, is that latent HIV infection is a disease of multiple tissues and very distinct compartments. And so this is a very challenging goal. Some of the data that has been published most recently, this is from Bob and Janet Silicano, shows that with a couple of different assays that we have to measure latency and the persistence of the virus in HIV, people living with HIV, what this is showing you is that over time, and now they have data up to people up to 25 to 30 years that have been on antiretroviral therapy, the size of the latent reservoir is basically the same. So that dotted line that you see in red is what was initially postulated, that if a person just took antiretroviral therapy for months or years, that the latent reservoir would eventually expire. And that goes back to the original hypothesis that these cells in red are going to have a half-life that's shorter than a person's lifetime. But what has become clear is that through a number of different assays, those cells are able to persist, and the virus is going to reactivate as soon as you take a person off antiretroviral therapy, even decades after they first encountered the virus. So I started this work in Hans-Peter Kiem's lab at the Fred Hutch, and I joined his group back in 2011. And at that time, a number of groups were just starting to study the first case of HIV cure in what was referred to as the Berlin patient, or Timothy Ray Brown. Many of you are probably aware of this case. He was a person living with HIV who also had an associated leukemia. And for that leukemia, he got a bone marrow transplant. And bone marrow transplants, as most of you are probably aware, are tricky enough to find a bone marrow match that is an allogeneic donor that you can accept cells from. But this Timothy Brown and some more recent patients, Adam Castier, who is known as the London patient, and some more recent reports from other parts of the world, all of these patients received a bone marrow or a cord blood source that had a mutation in a gene called CCR5. And you're gonna take the top. Oh, thank you. So the thing that all of these patients had in common is that they received cells that were mutated for CCR5, which is the co-receptor for most, but not all strains of HIV. And what they all have in common is when antiretroviral therapy was withdrawn, instead of the virus coming back, they were never able to find any trace of replication-competent virus as far as any of them have been studied. Tim passed away, unfortunately, a few years ago from a recurrence of his cancer. But to date, there's been no evidence that any of these patients have replication-competent virus in their body. This is in contrast to some other patients in Boston, Minneapolis, and there's a few other examples now that received bone marrow transplants that didn't have the CCR5 mutation, and they all rebounded after they stopped antiretroviral therapy. So this suggested right off the bat that the CCR5 mutation was causative for the cure in these patients. So when I joined the lab, what I was interested in doing is adapting this to a patient's own cells. So just very briefly, the idea here is that HIV in a human, or SIV or SHIV, which are two HIV-like viruses that we use in monkeys, use CD4 to bind a host cell, and then use CCR5 or sometimes alternatively CXCR4 to get into the cell. So what I tried to do as a postdoc was to use gene editing to basically recapitulate that CCR5 delta 32 mutation in the patient's own cells. So we're trying to make either a null mutation of CCR5 so it doesn't express at all, or we're at least trying to make a mutant of the protein so that it is not able to get to the cell surface where the virus needs it. So this is work that we are doing in parallel with a number of other non-human primate labs. This was really the sort of study that is tailor-made for the monkey model. So we wanted to understand each of the aspects that went into these cure cases and understand what the contribution of each was. So the first group that published here was Guido Silvestri and Leslie Keene, who at that time was at Emory, now in Boston. And what they did is they took hematopoietic stem cells out of a monkey. They then gave the animal total body irradiation, which is needed for the stem cells to engraft. And then they put those stem cells back in. And when they did that, and then they stopped antiretroviral therapy in these animals, the virus came back. So this suggested that just taking the cells out and giving them radiation and putting the cells back in wasn't going to cure you. And this is something that we recapitulated a couple of years later. And we went on to show that the problem here is that the radiation that is needed to make these stem cells engraft also kills some of the virus-specific T cells and B cells that are needed to fight the virus in the first place. So it's really a double-edged sword. We went further a few years later to show that with gene editing at that time, which wasn't highly efficient, we could get 5 to 10% CCR5 editing, and we could transplant those cells into animals. Unsurprisingly, that wasn't sufficient for cure in our monkey model, but it was really important for the next study I'll show you after this, which is where we looked at the allogeneic effect. And that's when we're taking donor cells from a different animal and putting them into our animal that's infected, in this case with SHIV. And the take home from all of this work is that this conditioning regimen at that time, total body radiation, our hypothesis is that it would deplete the reservoir just by killing infected cells. That was probably the case, but it was also taking out cells that were trained to find infected cells and were needed to participate in the fight. The allogeneic, what is referred to as the graft versus reservoir effect, that these cells from another person are going to see the infected cells preferentially and kill them. Again, there is evidence that that worked, but there was a lot of toxicities associated with that. So that wasn't something that we thought was tractable either. And that really left this idea of infection-resistant hematopoietic cells that has been one of my primary focuses ever since. So as I mentioned in this first story where we used gene editing, the amount of gene editing that we were able to introduce into these hematopoietic stem cells was fairly low, 5 to 10%. But it gave us enough data to perform a mathematical modeling study And we did this with Josh Schiffer and Fabian Cardozo Ojeda at the Fred Hutch. And this is a really valuable study and actually one of my favorites that I got to participate in. What they showed with their mathematical model, here looking on the y-axis is the CCR5 editing efficiency in stem cells, which at that time for us was anywhere between 40 to 60%. Nowadays, we're up to 90% or more. So that's improved quite a bit. And on the x-axis, what you're seeing is the ratio of the cells that we're putting into our animal model to the cells that stick around. So what you're seeing here in blue is what the criteria that you need to control infection. So what you're seeing here is that you need about 80% of your cells that are going in to be edited. And this is a log scale here. So what this is showing is you need about 10-fold more stem cells to go into the animal than are residual in the animal. In other words, there's going to be some stem cells that remain in your body. And if they're not edited, they're going to keep making T cells and other subsets that can become infected. So you have to replace them at least one log with the cells that you're putting in. These are both lofty goals. So we've managed to get our editing efficiency way up. So we've checked the box on this y-axis, but the x-axis is a lot harder because removing every stem cell from the body requires some toxic sorts of conditioning regimens that I'll touch on. And that's something that is going to be a high bar and probably not suitable for a person that's living with HIV that's otherwise healthy. So the take home then was that when I started as a postdoc, the focus was on playing better defense. And that was trying to replace almost, if not all, of the HIV susceptible cells in the body with gene edited cells that were resistant to infection. So what I'm going to talk about for the rest of the time is playing better offense. And that is reprogramming the host immune system so that they can find these infected cells, that they can go to all the different tissues where those latently infected cells hide out and actively clear them in addition to being protected so the virus doesn't spread. This obviously has to be safe and feasible. So I want to say up front that the stuff I'm going to talk about to you today is expensive. It's not in its current form feasible in the developing world. But what I'll touch on in parallel are our efforts to make this more feasible and make this a more scalable cure that can be applied in resource-limited settings. So all the cell types that I work with, or most of the cell types that I work with are summarized here. This is just a hematopoietic tree. So when I started in the lab, I was primarily focused on hematopoietic stem cells, which are at the top of the tree. So I'm showing you here in green just a generic gene cargo that we could introduce into hematopoietic stem cells. We can do this with viral vectors, namely lentiviral vectors, which are actually themselves based on HIV. We can use gene editing, as I mentioned, to knock out CCR5 or maybe other factors that the virus needs to replicate. And then my primary work was with hematopoietic stem cells, but I've broadened since then, and I'll touch today on some other stories that look at the same sorts of engineering strategies, directly modifying T cells, which are one of the more predominant cell types that are infected in the body, as well as B cells, which make antibodies that should be able to neutralize the virus, but the virus is very good at evading those antibodies. And so we have the opportunity with our gene therapy approaches to give these B cells a leg up. So this is how I'll divide the talk. I'll start with our hematopoietic stem cell work. Then I'll talk about CAR T cells and a couple of different stories within that realm. And then finish with in vivo delivery, which is how, again, in parallel, we're trying to make this all more democratized and available to a broader pool of people that are living with HIV. So I think this is always an important thing to highlight because I work with non-human primates. The first thing that I think most people know about non-human primates is that they're very expensive. These are obviously a lot more cost to take care of than humanized mice. They're harder to procure and source. And so we have to think very carefully about all of the monkey experiments that we do. We have to think very carefully about how we budget for our studies. And essentially, we try to gather as much data as we can in humanized mice and cell culture models, and then do our perfect experiment in monkeys. But once we get there, the monkey model has a lot of advantages, as you can see here. It's easier to sample. We can get more blood volume to look at more cells where we have these very rare latently infected cells that we're trying to find. We can look at all of the different organs, which can sometimes be difficult to characterize in small animal models. The animals live longer, so we can track over years if the cure that we're able to introduce into the animal is stable, or if the virus comes back months or years later. There are other practical aspects. So these are bigger animals. And so in thinking about scaling up to a human, it's a nice sort of mid-range from going to something that works really well in humanized mice and thinking about how you're going to have to scale that up for a person. Thinking about the amount of vector, the gene editing nucleases. I'll talk a little about nanoparticles today. That's a scale-up question that we've had to deal with as well. Genetic distance to human is obviously quite close, closer than it is in mouse models. And I think what's really important here is there's an intact immune system. So the immune response to some of the gene therapies I'm going to talk about isn't necessarily toxic, but it can downregulate how efficacious they are. So this gives us an idea in an immune competent model, just like a person, how well these therapies are going to work. Yeah. So we usually don't go smaller than 3 kilograms. 3 kilograms is about the smallest that we would go. Depending on the animal, they can be up to 20 kilograms. 20 kilograms gets really challenging because a lot of our doses are weight-based. So in a perfect scenario, we're somewhere between 5 and 10. Hmm? Oh, sure. I never know how to do that either.
That's weird.
Yeah. You're welcome. So yeah, talking about our stem cell work. I work in, I think as many of you do, I work among a lot of cancer biologists and virologists. So we try to think about solutions that gather as much from the cancer field as from the HIV field, because there's a lot in common. If you have a cancer and you're in remission from your cancer, the first thing that you have to think about is durability. You're cured of your cancer now, but are you going to be cured for the rest of your life? And I think that's going to be where we go for HIV. When we eventually get to a point where people are cured, there's always going to be a question. If I'm not taking my antiretrovirals anymore, could the virus come back months or years later? So we want something that we can feel confident is going to be durable for the lifetime of the individual. So I'm schematizing this here with some data that I'm going to talk more about later, and that is with CD20 CAR T cells. So this is a CAR T cell that is being used right now in people that have B-cell leukemias that overexpress the CD20 B-cell molecule. We can make these same CAR T cells from monkeys and show that they expand quite well. So in this example, you're seeing that they're expanding up to 50% of the total T cell compartment in peripheral blood has this CD20 molecule. But what you can also see here is that the functional period of these cells is very short, a month or two at most. And then after that, they basically go away and we never see them come back again and B cells come back. Now, I showed this on this very long x-axis to contrast what we know about HIV rebound. So those patients from Boston that I highlighted at the top that didn't receive the CCR5 mutated cells, they rebounded 3 to 8 months after ART was withdrawn. Many of you have probably also heard from a long time, several years ago now, the story of the Mississippi baby who was electively taken off antiretrovirals shortly after birth. And HIV didn't rebound for 2 years in that child after ART was stopped. So this is a very long time frame. And so we have to get our cell-based therapies to the point where even if the virus comes back months or years later, we're still going to be able to have these cells wake back up, find the virus, and knock it down. So the first story that I want to highlight, I think, is one that you all know quite well. This is work that we did with Scott and Angie. This is one of my first collaborations that I had as a postdoc, and I still value it quite a bit. I had a lot of fun with Scott and Angie and also Jerry at that time working on this project. So you've heard the story about introducing introducing HIV-specific CAR molecules in hematopoietic stem cells. And the take home from that I won't get into today is that they did persist lifelong. So this was really the proof of principle to show that this is a really great way to get our anti-HIV molecules to express for the lifetime of a monkey in this particular study and have persistent function throughout that time. Talking about tissue trafficking, this is another thing, obviously, that's in common for HIV and cancer. If you have a solid tumor, you have to have your cells go into that solid tumor and still function. That's a huge part of immunotherapy right now for cancer, since the tumor seems to be so good at turning those cells off. And we have similar concerns about HIV. So what we did is we followed up with those same monkeys from that study with Scott and Angie to ask questions about how they traffic. So this is one of the important tricks for working with monkeys, is you try to get as much data and as many reports out of a single study as you can, just to make the most value and really have the most reverence for the animals that we're using in these studies. So we use the same animals, and what we showed here is that the CAR molecule is actually really well expressed in some of the tissues where we know that HIV persists. And this is specifically in B cell follicles and lymph nodes. So I'll talk more about the B cell follicle and our other attempts to target this. But what we got out of this particular study that was done by Isaac and his wife, Valerie, who were in mine and Hans Peter's group several years ago, is that the stem cell-based CAR T cells and other CAR expressing subsets very efficiently traffic to these sites where HIV hides out. So then the last thing that we were able to address with the stem cell studies is this idea of toxicity versus efficacy. And this is especially an especially important question for hematopoietic stem cells, because we have to go to sort of an extra link to get these cells to engraft. And this gets into the work, that we're doing right now. So this is led by Teresa, who's a senior technician in my group, and Jason, who's a postdoc working with me and as well as with Hans Peter. So just to show you how these experiments work, we can collect CD34 positive hematopoietic stem cells from the monkeys. There's two species of monkeys that we use in our studies. We use rhesus macaques and pigtail macaques, and there's some certain details I won't get into today how we pick the species, but we can go back and forth between those two. We can then culture those cells in a dish. We can modify them with lentiviral vectors and gene editing nucleases and other vector types that we can use to modify any gene of interest. And then that gives us a stem cell product that we're ready to put back into that same animal. The major limitation for hematopoietic stem cell gene therapy is that if we just put those cells into an animal, they're not really going to engraft and persist. We have to quote unquote make room for them to stick around. And the way that we make room is with a conditioning regimen. So I mentioned at the top, total body irradiation is what we historically have used quite a bit. As you can imagine, that's quite toxic. It makes plenty of room for the stem cells and they engraft quite well, but it also takes out some virus-specific immune cells that we don't want to lose. And so since then, we've really moved on from total body irradiation, and now our sort of standard for comparison are alkylating chemotherapies. So these are small molecules that will intercalate into DNA and or block DNA replication. So cells basically die when they're trying to divide in the presence of these alkylating chemotherapies. Where we want to go is something that's even safer, and that is using what we refer to as ADCs, or antibody drug conjugates. So this is an antibody that we can target to any cell type. What you're seeing here is CD45, which is on the surface of most, if not all, hematopoietic cells. So if we want to just clear out the hematopoietic system, for example, without having as much toxicity on organs, we can put in CD45 ADC. And these antibodies are conjugated with these little yellow blasts here with radiation. We can put radioisotopes that will only kill the cell that's right next to it because they're very weak radioisotopes. We can put on transcription inhibitors or translation inhibitors, all kinds of different things that will kill just the cell where that antibody binds. So the goal here is to really refine how we're making space for these stem cells to engraft while letting those stem cells engraft just as efficiently as with the more toxic regimens. So we put this back into the animal, and then we follow these animals over time. So this is an example of the data that we get. In the inset, what you're seeing is with our CRISPR editing strategies, the efficiencies that we're able to get. So the way that this works is we collect the cells on the first day. On the second day, we edit them. And on the third day, we put them back in. When we put them back in, the editing is still going on. So what you can see is that the animals here that were conditioned with any of these different conditioning sources, we have about 50% editing on the day they go in. But when we let those cells continue to edit in a dish, which we presume is also going on in the animal, we're getting upwards of 90% or more editing. So the editing works quite well. And what that translates to in the animal is shown here. So in the black is our busulfan conditioned animal. So after we collect the cells that we put in busulfan, and here we're comparing that to two ADCs, either CD117 which is hematopoietic stem cell marker, or CD45, which is a more pan hematopoietic marker. And you can see that busulfan does the best. We want these ADCs to be better. We know that they're less toxic, but this is one of the tricks that we're currently working on, is that oftentimes less toxic means less effective. And that's what we saw in this particular example. The good news is that with busulfan, at least, we're now able to get up to about 40% of the hematopoietic system to be edited at CCR5. So this is the highest that we've seen so far. For CCR5, I don't think anyone's gotten higher. Other targets can sometimes be edited higher, but we think that's a locus-specific effect. So the take home here is we're on the right track, but we're still looking for ways to get these edited cells to engraft more efficiently without having the toxicity, in this case, that we see with the busulfan conditioning regimen. Where we're going is that instead of just editing the locus, which is shown here on the left, so when we use CRISPR to edit a targeted locus, usually that's by non-homologous end joining. That forms insertions and deletions, or indels, which abrogate the function of the gene. And this is what we would go back to as better defense. So the data that I just showed you is only editing CCR5 and inactivating it. But what we want to do to play better offense is insert something at that edited locus with a viral vector oftentimes, so that we target it directly to that site with homology on either end, as is shown here in yellow, and then that will insert into the targeted locus. And that's where we'd like our gene editing approaches to go. There's also limitations here. These are actually pretty well known. I'm showing you a series of papers that some are over a decade old now. And the common theme is that the better you are at inserting a sequence into a locus in a dish, in this case from Matt Porteous, he was approaching 100% efficiency of inserting a GFP transgene into this targeted locus, the worse those cells are at engrafting in the animal. So this is something that's been shown over and over in mouse models. And I'll show you here, we see the same thing in monkeys. So in this experiment, we had two animals, and what we did is we divided those stem cells that went in each animal into 4 pots. And each of those pots are shown here in blue dotted lines or orange dotted lines. And what we did is we edited either at CCR5 or another locus to prove that there wasn't any locus-specific effects to why these cells were and weren't engrafting. And then in half of the CRISPR for each, we tried to insert just a little oligo. Nothing big, just a little sequence at that targeted locus. When we put all of these cells into the animal, what you can see is that when we just edit the locus with CRISPR, those cells engraft quite well. In this case, around 20 to 30% in the first animal that had a higher cell dose, and they engrafted less well, but this animal also had a very low cell dose. But in both cases, what you can see is when we try to insert just a little oligo into either the CCR5 locus or this other unrelated locus, HBG, those cells don't engraft hardly at all. They become very hard to find over time. We don't know why this is. There's a lot of hypotheses in the field about what could be going on here, but the take home right now is that when we try to add on to a gene editing approach by inserting something, our stem cells don't like that. So these are really the two main challenges that we have that we're working on right now. What we've shown with our hematopoietic stem cell work is that we can use our monkey model to explain what happened in these cure cases and understand what was important and not important. But what we need to do better is figure out how to engineer these cells in a way that they will engraft and function in vivo. And as I'll talk about in the next few sections, understand how to do this in a more scalable and economical way so that we're not having to take these cells out. We don't need these very expensive facilities to engineer them. We can just give a person, hopefully, a shot, and all of these engineering strategies will go straight to the cells in the body where they need to work. So I'm going to change gears. And this is sort of the work that started to grow me into independence within Hans Peter's lab. So this is all stuff that I started working with him, and I'm now carrying on in my own independent laboratory. So this is T cells instead of hematopoietic stem cells. And the big advantage with T cells is that the toxicity is a lot lower. So what I'm showing you here is what you might find familiar from a few slides ago. The same as with our stem cell approach, we can collect T cells in this case from these animals. We can do it either from uninfected animals or where we're modeling our suppressed HIV infection. Again, we culture them in a dish and engineer them with all the different gene therapy tools that we have in our toolbox. The big advantage for T cells is that conditioning we feel is optional. We've done some studies with a low level of conditioning, and even with a low level of conditioning, there can be some tricky aspects to this. We've done others where we've done no conditioning, and the cells have worked quite well. I think it's still a lot to be worked out about how important conditioning is for T cells, but at the end of the day, it's going to be less toxic and less impactful than it is in the hematopoietic stem cell setting. And then again, once we've made our product and we have or haven't conditioned the animal, we put those cells back into the autologous host. This is just a schematic of how we make the CAR T cells, and without belaboring the point, basically we're taking T cells out of the body. We're using viral vectors that encode these CAR molecules. We put those into the T cells, grow them to quite a large scale, and then put them back into the body. This schematic is focusing on cancer, and that's where these CAR T cells have really worked the best so far. So we have FDA-approved CAR T cells now that target B-cell leukemias that overexpress CD19, CD20, as well as BCMA. And what we'd like to do eventually is get to the point where the same technology can be used to target the virus. And specifically, we want to target the envelope protein, which is expressed at the surface of an infected cell, and we think is the best marker for CAR T cells to target. Where I'm going to start, though, before I get to the HIV CAR T cells, is go back to that data that I showed you earlier. We had our CD20 CAR T cells that worked quite well. When we were first starting to develop our CAR T cell model in monkeys, it was very challenging. And I still remember going to a conference where Leslie Keane presented the data that's in this paper, where they had just gotten CAR T cells to work in a non-human primate model, and they were using CD20 at that time. So I want to highlight these efforts from Leslie Keane and Mike Hutchinson. Leslie at this time was in Seattle, now she's in Boston. And we grew out of their first report of this in the non-human primate model. So as I showed you earlier, durability of the CAR T cells when we're just putting adoptively transferred T cells is a weakness for the T cells. As of right now, we don't have any evidence that these T cells are going to last forever. It's not even clear in patients, to my knowledge, that they last forever. They hang out, but their function, I think, still needs to be understood a little better. But what CAR T cells and adoptively transferred T cells more generally can do, we hope, is efficiently traffic to tissues and have a better toxicity profile. In other words, do the same job without the same toxicities that we're having to negotiate in our hematopoietic stem cell experiments. So the CD20 CAR T cell story was important for us for two reasons. First was to benchmark how well we can get CAR T cells to work in the monkey. So this essentially becomes a positive control. If we can get these CD20 CAR T cells to work, then we suggest that we can get our HIV CAR T cells to work as well. The other part is that it actually has an interesting biological aspect to trying to cure people that are living with HIV. This goes into one of the specific tissues where HIV hides out, and that's in lymph nodes and spleen, B-cell follicles that are found in multiple secondary lymphoid tissues. So what I'm showing you here are these rare infected cells, which are T follicular helper cells in yellow. And what's known is that these B-cell follicles are a site of B-cell training. So we don't want a lot of T cells in the mix while these B cells are figuring out whether they're going to persist or not. So for the most part, CD8 T cells are excluded from the space. And that allows these infected cells to persist and hide out among these nascent B cells in the follicle. And this is all work I'm highlighting from a number of different groups, Liz Connick, Pam Skinner, and Offman and Lewis at Oregon Health Sciences University. So in our study, what we wanted to do is start with an animal that had this latent infection in B-cell follicles. We wanted to disrupt that follicle, and to do that, we used the CD20 CAR T cells. Then we wanted to reform that follicle, and the hope was that while the B-cell follicle was disrupted, these CD8s could get into the space, see these latently infected CD4s, kill them, and then when the follicle reforms, those latently infected cells are gone. So this is work that's being done by John, who's an MD fellow working with me, and Hans Peter, and Carly, who's a staff scientist in my group. So I showed you earlier the graph that these CD20 CAR T cells expand quite well. What I'm showing you here is how well they function, and that's in knocking down CD20 positive B cells. So this is not a cancer model. These animals don't have B-cell leukemia. So we're putting these CAR T cells, and we're just looking for what we refer to as B-cell aplasia, and that's knocking down just the healthy, any B cell that's in the animal. And you can see that works quite well. So this is in animals in black, that were uninfected, and then in our ART-suppressed infection model in red. You can see that we knock them down to undetectable levels for a defined period. The uninfected animals, their B cell counts rebound quicker. They're a lot sluggish, a lot more sluggish to come back in our infected and suppressed model, but it's working in both cases. The other thing that we're able to develop that was really rooted in the work that Mike and Leslie Keene did is to manage their cytokine release syndrome symptoms. So as many of you know, when a person gets CAR T cells for cancer, these T cells work so well that they're spitting out a lot of cytokines, driving fevers, and you feel really bad, and people can get really sick. And there's a number of ways that we can control that so that the CAR T cells are still working, but without so much toxicity. And that's using tocilizumab, which is an anti-IL-6 antibody here in orange. Steroids, in our case, we use dexamethasone up to twice a day, and gray and yellow, something as simple as Benadryl. And then we're dosing those as we monitor in the serum IL-6 and C-reactive protein, or CRP. So this is just showing that when we can get these readouts in the blue and green lines, so that we can then dose with our IL-6 blockade or steroids to knock that down, but the T cells are still able to work. I'm highlighting down here in the lower right, anakinra, which is an anti-IL-1 receptor antibody, is something that, excuse me, we're also using specifically for neurotoxicity that often also happens with these CAR T cell therapies. I want to highlight, this is one of my favorite parts of working with Hans Peter. He's a clinician and a very well-established clinician in the hematopoietic stem cell space, as well as in the CAR T cell space. So all of the CRS prophylaxis that we developed here was in close collaboration with him to apply the same practices that they're using in the clinic to our monkey model. So after we get these CAR T cells to work, we want to ask the question of how well they targeted the viral reservoir in all of these different tissues. So these are very eventful days that we have. So in an animal in study for any of our non-human primate projects, we work very closely with the Primate Center and Bob Murnane, who's the senior veterinary pathologist at the Primate Center, to collect 25 or more tissues from numerous sites throughout the body. Most valuably for what we're doing is the brain. The brain is obviously something that we can't get until end of study, and we think that the virus hides out there as well. So we take different portions from the central nervous system, as well as all of these secondary lymphoid tissues and other relevant organs. So this is just one example of what we do with those tissues, and this shows you how well these CD20 CAR T cells work in these sites. So I showed you earlier that with Scott and Angie, we were able to show that our HIV CAR modified cells traffic to these lymphoid follicles, these B cell follicles. So these are from our CD20 CAR T cell study, peripheral lymph node, either axillary or inguinal, mesenteric lymph node or spleen sections that were taken 2 weeks after CAR T cell, CD20 CAR T cells, 4 weeks, or in this case, over a year later. So going right to left, what you can see is after a year, when the CAR T cells have contracted and B cell counts have come back, our B cell follicles reform quite nicely. So that's shown here in brown. But as you can see on the left and in the middle, at these acute time points after the CAR T cells go in, these follicles are basically non-existent. We're doing a very good job of knocking them out, which was really gratifying because not all of these CAR T cells for cancer work very well in tissues. CD20 seems to be, in our experience at least so far, an exception to that rule. So what Carly did after generating that IHC data to show that the B cell follicles were gone, the next part of our hypothesis was that the TFH cells that are infected with the virus, they should go away too. And she does show that here. So this is just her flow tree for looking at TFH in these lymph node follicles, and this is the quantitation of that data. So at that 1 month time point, that middle slice in that IHC data that I just showed you, we had limited samples available, but from the 4 samples we did, it was very difficult to find any TFH in the lymph nodes that we took at that acute time point. But at those further out time points in these animals, the previous slide showed you over a year, it was anywhere from 6 to 13 months on average, these TFH cells come back. So they are reconstituting these space, but the question is, did we actually get rid of the infected cells? And unfortunately, the answer is no. So what I'm showing you here on the left is PBMC that we took at these serial time points, including before the CAR T cells went in, the day that the CAR T cells went in. These are the 2 tissue surgeries where we also took blood, and then this final necropsy time point. And this is looking at, if I, here, maybe I can't do that. Yeah, no. So the viral DNA that you're showing here on the y-axis, so we're just measuring total viral DNA. Those of you who are involved in these cure studies know that this isn't the most precise way to measure the viral reservoir, but it's sort of a quick way and gives us a yes or no answer. In this case, the answer was no. We're seeing maybe some reduction over time, which we're not sure is related to the CAR T cell therapy or not. But overall, the levels of the virus are very consistent. When we actually look in the different tissues that we collected these 2 tissue time points and also a necropsy, it's kind of a mixed bag. In some tissues, it does look like we're seeing a trend to a reduction in the size of the reservoir that does reach statistical significance in the spleen. But other compartments, including the gut and bone marrow, there's really no change at all. So as with a lot of the strategies we do for HIV cure, the take home is that we're making a dent in the reservoir, but we're not getting anywhere close probably to cure. And getting back to these very extensive experiments that we do with all of these tissues collected in necropsy, that's shown here. So the blue, orange, green, and yellow are the 4 SHIV-infected ART-suppressed animals from our suppressed infection model, and gray are some historical controls that we're comparing against. As you look across all these different compartments, you can see some where it seems like we went down, maybe others where it seems like we went up. This is at the DNA level. This is looking at viral RNA. And again, it's really a mixed bag. So the take home from this is that we can show that these CD20 CAR T cells work. This is a very important comparative example to show that we can get generally CAR T cells to work in the monkey model, that there's not something about monkeys that CAR T cells generally don't work, which is an important thing to establish. And we made this interesting note that B cell recovery seems to be lesser in our ART-suppressed infection model, which from conversations with clinicians is something that I think is starting to be observed in people living with HIV that receive B cell directed CAR T cells as well. But what we found is that ablating these B cell follicles and redistributing these TFH is not going to be sufficient to impact viral latency. So the thought then is whether we can combine this with something else that's going to be more potent. So there are 2 examples here where we have or are continuing to look into this. With OFFAM at Oregon, we're repurposing CAR T cells to target other innate immune subsets, which may or may not be involved in contributing to the persistence of the virus. And with Larry Corey at the Fred Hutch, Larry is just about to publish a paper that we helped with where PD-1 was targeted on the CAR T cells as a marker for latently infected cells. And that's an interesting story that should be coming out soon. Coming back to this question, what are we doing to try to make this better? We're coming back to our virus-specific CAR T cells. And I haven't gotten into how these work so far, so I wanted to briefly talk through this. So these are called CD4 CAR T cells. That's a confusing name. We didn't name that. This is how the HIV-specific CAR molecule was first named back in the early 1990s. This was the first chimeric antigen receptor that was described in the literature. And it's called CD4 CAR because it literally uses CD4 to recognize the virus. So this is another schematic showing that a virus binds CD4 and CCR5. It docks at the cell surface. It injects its genetic material into the cell, reverse transcribes, and integrates into the genome. So now this is this persistently infected cell that we're trying to target. So from that integrated provirus, more viral RNA is made, more viral proteins are made. And the key protein that we're trying to target is envelope, which based on a lot of historical data is out there, should be one of the first proteins to arrive at the cell surface so that our immune effector cells can see it. That then allows the virus to assemble at these sites and make another infectious virus that can repeat the cycle. So our CD4 CAR T cell literally uses the same CD4 moiety that's at the cell surface and binds envelope and allows this virus to get into the cell and uses that against it. So it's become pretty clear that a virus, an HIV virus that can't use CD4 is really not going to be fit to effectively replicate. So we're exploiting really a fundamental requirement for viral replication in the CAR that we're using. The problem is that HIV is a lot harder for the immune system to see for a number of reasons. And one of those is just a numbers game. So the story that I just told you was CD20. A person that has cancer, by definition, is going to have a lot of these these leukemic B cells. And on a per cell basis, the amount of CD20 that is expressed on a cell is very high. This makes it a lot easier for this CAR T cell to see it and to kill it. In contrast, a person that's living with HIV that's taking their antiretrovirals every day is going to have very few of these latently infected cells. It's thought to be 1 in a million CD4 T cells or less. And we honestly don't know exactly how many envelope molecules are expressed at the surface of these latently infected cells. It might be 0, but it's very hard to measure in vivo. So the way that we tried to get around this is to provide some more cell-associated envelope antigen that will keep these CAR T cells happy and keep them going while they're sort of muddling through this tough job of finding these very rare cells that have very low levels of antigen. And this is work that we started with Jim Riley and Amy Payne. Jim's at UPenn, Amy's recently moved to Columbia. So I won't go through this. This was published a few years ago, but this was sort of our pilot study showing proof of principle that this could work. And to make a long story short, we had a K562 cell line that stably expressed envelope. We irradiated that cell line and put it into these animals and showed that the CD4 CAR T cells that we previously put in could expand. And then when we took ART away and the virus rebounded, we were able to see stable virus remission in 2 out of these 4 animals. This wasn't a cure. This was a reduction in the amount of virus, and we'd see little blips over time. But this was the first measurable impact that we'd seen in any of our virus-specific T cell studies. Going forward, I'm not going to go through all the data that we generated before and after this particular study, but this turns out to be a very challenging problem. So the 2 animals that I'm showing you right here are the only 2 that we've been able to get this to work so far. So we think that we're on a knife's edge again of efficacy for how these therapies work. And I'm grateful to have now funding that will give me 5 years to figure this out. The way that this grant was put together was in the first aim to focus on this antigen boost. I mentioned that we were using K562 cells that stably express this antigen. An immortalized transformed cell line is not going to be something that we can use in a patient. So this is developing better ways to qualify what a good boosting antigen is and figure out the best way to deliver it. The second aim is to further develop the CAR molecule and the CAR T cells. So what we proposed in the grant was to use CRISPR to knock out host factors that could inhibit these CAR T cells function. For some technical reasons, that's not, it's seeming like that might be a dead end. But so where we're now going is looking at different domains and how we can swap domains within the CAR to make them work better. And then this is usually the theme with our monkey grants. The third aim is always testing them in the monkey model that I've already shown you today. So that leads me to where I'm ending up here with the in vivo delivery approach and specifically thinking about how we're going to deliver this envelope antigen to boost these CAR T cells. So the in vivo delivery question is quite a challenge. For gene and cell therapies, this is where everyone eventually wants to go. The obvious huge criticism for gene and cell therapy right now is it's very expensive and really limited to resource high countries like the United States and Northern Europe. So what we want to get to a point is where we can have a therapy that's safe, that's portable, that works, in our case, achieves viral remission, will last lifelong, as I've already talked about, is cheap, and that if needed, we can repeat over and over again and redose as needed. So in the case of our envelope immunogen to stimulate the CAR T cells, this is sort of a Maslow's hierarchy that I put together for that grant to prioritize how we would do that. And there's 4 levels to this. The first level is to make an envelope that expresses very well at the cell surface. This is what the CAR T cells are going to be able to see, and we want to make it easy for the CAR T cells to see it. So this is work that we're doing with Jim Hoxie at the University of Pennsylvania, who's developed a number of immunogens for vaccines that he already has shown in this data here express at very high levels. So CPMAC and Delta GY are 2 examples of ONs that you can see compared to the parental envelopes at the top, have higher levels of expression and almost, in most cases here, coat the entire surface of the cell. So John in my group has been working with Jim to image these using a really cool imaging flow cytometer and grade them based on that. The next level of the pyramid is obviously that we want these envelope immunogens to be able to stimulate the CAR T cells. That's really the whole point. So Nikita, who's a former technician in my lab now at grad school at UW, developed this assay where we transfect our candidate envelope mutants, mRNA, using electroporation into a cell line. Then we put our CAR T cells on top of those cells and load them with a cell trace dye that will dilute as the cell divides. So the more dilution we have in the signal, the more we know that they're dividing. And when we look at the whole panel of envelope mutants that we got from Jim, you can see that all of these mutants work very well. So we have a negative control over here, which is a delta D mutant that Jim has that shouldn't bind CD4 and therefore shouldn't bind our CAR. So this is sort of the baseline that we see. This is our on that was in that K562 cell line. And these are all the different mRNA mutants that we compared. And they all seem to outperform what was in the K562 cell line. But whether or not these are better than each other is kind of hard to figure out. So the next two I won't get into for the purposes of today, but the other things that we're looking into are that these aren't inhibited by anti-envelope antibodies, which is stuff that we'll get to. And the top of the pyramid is serial killing, meaning that these CAR T cells can see a target, kill it, and move on to the next one and be able to persist over time. So I'll skip ahead in the interest of time, but the other part of this grant that we're really excited about is to look at different CAR molecules in the same animal. So this is another advantage of the monkey model. What we can do is in this particular experiment, which is being led by Lucy, who's a PhD student with me and Hans Peter, and being supported by Keith Jerome, a PI at the Hutch who really specializes in molecular virology assays. We can build all of these different CAR T cells, which, as you can see here, have different co-stimulatory domains and transmembrane domains, just shown by these different colors. And this particular experiment, we're going to have up to 20 different CAR T cell products that we put into the same animal. And with Keith, we can then use PCR-based assays to distinguish each one. The hypothesis is that if we can compare them over time, some of them are going to expand really quick into high levels, but then they're going to drop off. Some are going to expand to lower levels and then drop off. And some, like this one in orange here, is what we're looking for, where it has a nice expansion and then it persists over time. So the hypothesis is that we're going to be able to find the best organization of domains that allows these CAR T cells to function persistently over a longer time period and more durably. We're modeling this using our CD20, which is why that CD20 story is so important. And then the best domain structure that we find with CD20, the goal is to move that over to our CD4 CAR for HIV. Big picture, though, where we want to go back here is to in vivo delivery. So I've told you is that in your right arm in this example, we could have an envelope mRNA that's an untargeted LNP. So that's going to stimulate our CAR T cells. And then your left arm, you could get LNPs that are encoding our CD4 CAR molecule. So LNPs are lipid nanoparticles. Again, I'm very fortunate to work with Drew, who just won the Nobel Prize for his LNP work. We've obviously had, I think everyone in this room, proof that LNPs are safe and very effective. And so Drew is now working with me and a number of other groups in a number of different disease settings to even broaden how this nanoparticle can be used. And in our case, we want to deliver the CAR with it, and we want to deliver the envelope mRNA to stimulate that CAR and be able to redose over time every time that it's needed. The last, how am I on time? I'm good. Okay, yeah, I'll be very brief here, just in the interest of time. The last thing that I want to touch on is our B cell in vivo delivery. What we're doing here is trying to edit B cells, and we're trying to modify them so they express any antibody of interest. So this is work that was first published by Justin Taylor, who was at the Fred Hutch and is now at the University of Virginia. What he was able to develop is what's called an EMAB, or an engineered monoclonal antibody sequence, that is a fused light and heavy chain that inserts right in this region of the immunoglobulin heavy chain, or IgH locus, and it essentially reprograms B cell specificity in that specific T cell. It also features this tag linker, which forces this heavy chain and light chain to pair and can be used for detection of the modified B cell. We're pairing that with work that we do with Hildegard Boining at the University of Hanover in Germany. What we've done with her, and this was Bill and Melinda Gates Foundation funded work, where we identified AAVs that are specific for a specific cell type, in this case, B cells. So with Hildegard, we can put in a library of mutagenized AAV capsids. So this is a vector that has tropism for different cell types, and we can find the capsids that are most specific for B cells. So we use B cell specific AAVs. They're novelly engineered with Hildegard, and then we package them with this CRISPR editing machinery that was developed with Justin. So this is work that I'm doing with Anne-Sophie, who's a senior staff scientist in Hans Peter's group. And what she has shown is that qualitatively, she's able to knock in this case, VRC01, which is a broadly neutralizing antibody for HIV, into the edited IgH locus in monkey cells. And this is just a gel-based assay to show that qualitatively she can do that. But this is using that tag linker that I mentioned. So by flow, we can actually see that this antibody expresses at the surface of our edited B cells up to 10%. I think what's most relevant for this audience is a historic paper from David Baltimore that we referred to quite a bit, which suggests that we really probably don't need a huge efficiency of B cell editing to get a therapeutic effect. Because B cells are such efficient protein production machines, if we can edit for what was postulated in this paper, maybe just as little as 10%, which is what we got here, then that would be enough to have a therapeutic effect in an HIV setting. We're not obviously to that place yet, but this is really nice and exciting proof of principle as we head into mouse and also monkey studies to test this approach. This is just showing how we make these vectors. I think one thing that often comes up for those of you that think a lot about HIV is that AAVs are relatively small. You can't package a lot of material. So for our CRISPR machine, we have to divide that into 2 different vectors. But the other trick about what Hildegard does is that she has a chemistry approach that allows us to link these 2 vectors. So they basically drag each other into the same cell. So we're essentially doubling the cargo capacity. So we call this non-immunogenic because we're pulling this out in immunocompetent animals where the immunogenic capsids should be cleared. Cargo enhanced, or NICE AAV is our quote unquote clever name that we came up for this system. So I'm going to stop there and just wrap up by saying we want to get more broad with our B cell gene editing, as with everything. This is just a report that talks about sort of the delay in getting these broadly neutralizing antibodies to penetrate to these different tissue sites where they're needed, in this case for virus acquisition. We think B cells are going to do a great job of that. But beyond that, this is my last slide just to say that B cells are really the next frontier. What I've shown you today is that HIV cure has a proven track record with stem cells, with the Berlin patient and others. The toxicity of the conditioning regimen is something we have to take into account, though. T cells has an established path to the clinic for cancer, but we have to deal with the issues of persistence and low antigen diseases like HIV. And B cells is very exciting, but I only gave you about 5 minutes of that today, and that's because we're very early in that process, and there's still a lot to learn about what the best way to do that. So I will stop there. I'd like to thank my brand new lab with this picture that we just took last week. This is all the people that are doing the work. Also want to highlight our non-human primate team that's on site at the University of Washington nearby that does all the hands-on monkey work. Our collaborators here at UCLA and elsewhere at the Primate Center, and all the people from the Keem Lab, as well as funding. Thank you very much.
Thank you, Chris. This is an amazing amount of data. Impressive. Do you have any questions from audience?
So for gene editing, you sometimes use RNP for customizing, sometimes use RNP. Do you have any preference?
If we can use an RNP always works better.
Oh.
It doesn't always package well, for example, in lipid nanoparticles. So for lipid nanoparticles, we have to use mRNA because I think just the chemistry and the biophysics of those nanoparticles, those ribonuclear protein complexes don't package well. There's other nanoparticle that, for example, plug Ginader at the Fred Hutch uses gold nanoparticles. There you can decorate it with RNPs and other nucleic acids. So it depends on your platform, but RNPs are better if you have a choice.
When you deliver Cas9, don't you deliver gRNA together? I mean, in the same RNP?
Yeah. If it's an RNP, you pre-incubate them for like 10 minutes on the benchtop, and then you electroporate.
for your AAV approach, you express the gRNA from a different AAV.
Yeah.
And Cas9 from another.
Right.
So why did you choose to do
that vectorization? Well, more or less, it's because Cas9 is so big, we can't fit anything else on that vector. There are smaller Cas9s that are available, so that's an option we have. I don't disagree that what we did here might need to be changed in some way, but this is where we're going to start, and then we'll adjust as we need.
In the CD20 study, I'm just trying to recapitulate that with the CD20 core with our previous studies with peripheral HIV specific.
Yeah.
And basically, we've got 2 monkeys that responded to the vaccine.
Mm-hmm.
But what we didn't see, in at least our monkeys, and I imagine Riley's monkeys, other monkeys, is that you didn't see migration of those cells to molecular.
Correct.
I mean, we looked and we saw, you know, and Myra was just publishing this paper that we saw limited persistence, limited graphic in lymph nodes.
Mm-hmm.
Do you think the antigen CD20 is required to be really helpful in getting those cells in there? And you know, with HIV, I mean, there's a lot of confounding factors. If you have higher levels of antigen in HIV, with the cells theoretically into the body.
Yeah, it's hard to say. And yes, to confirm, I mean, you know that paper in our more recent IHC, we do not see those adoptively transferred CAR T cells go into tissues the way the stem cell-based CARs do, for sure. It's black and white. What's happening with CD20, I think there's 2 variables. It could be something about CD20 specifically. Maybe that's just a better target that a CAR T cell can find in tissues. The other variable is this isn't a cancer model. So there could be tumor-specific things that in a tumor setting, CAR T cells are being more actively turned off than they are in our healthy animals. So that could be a big difference as well.
I mean, I've had this discussion with Jim Riley. I think the antigen levels.
For sure. I think above all else, I think the model for what we saw in that paper is that latently infected cells are coming out of the tissues to blood where we know the CAR T cells are working, but we don't have evidence that those adoptively transferred cells are getting in.
I just want to ask about the gene editing for the HSCs.
Mm-hmm.
It's kind of striking to see the transplant efficiency. Have there been any see other types of genes that were done to see what's the difference of the transplant?
I would really love to do that. I don't have the money, but I would really love to do that. I think that's a single cell. I feel like that's a single cell because you really want to drill down to cells that have the persistent phenotype. So that becomes very costly. But I think some of the hypotheses out there is it's a DNA damage response that's blocking engraftment. It could be that the cells where we get integration are cell cycling, and you want a quiescent cell to engraft. Those are the 2 models that we've thought most about, but it's been a really tough problem to track down. And there's this company, Graphite Bio, that Matt Porteous had where, they had problems. And I think the data that we had is consistent with the problems they had. So to my knowledge, no one knows the solution right now, but I think whoever figures it out is going to be very well off.
Same line of question. I thought the homologous recombination is very low efficiency in quiescent cells.
Right.
I remember Paul Cannon come up with some kind of compound, chemical.
I-53.
Yeah.
Yeah.
Does it really work?
We haven't gotten it to work in our hands, but we've also shown in a paper we put out a few years ago that RNase activity in true hematopoietic stem cells is relatively high. So the I-53, as well as the zinc finger nucleases that we used to use for editing, were introduced by mRNA. And we showed that if we actually put an RNase inhibitor in with the mRNA, we could increase expression of the nuclease and get better editing. I think that the I-53 didn't work for us because the mRNA was getting chewed up. We tried to make the peptide, and that was kind of tricky too. So I can't say whether or not we did it optimally, but it didn't work in the hands.
Any other questions? Okay, thanks everyone.
Thank you.
Another comment from the chat, great talk. Thank you.
Thank you.
Thank you.